Welcome to Shalu's Biology. In today's lecture, we are going to study about polymerase chain reaction. Suppose you have a small segment of DNA molecule and you want to detect it or you want to research on it. But finding a single gene out of thousands of genes is like finding a needle in a haystack. What can you do? You can make identical copy of this gene of interest, thousands of copies, so that you can easily detect it and you can study about it. The next question you are thinking about is probably how can you amplify or you can make identical copy of your gene of interest. What the scientists did previously is they integrate their gene of interest into the bacterial plasmid, and that bacterial plasmid, when insert into the cell, bacteria cell replicate and make thousands of copies of your gene of interest. But that method was very time consuming and laborious. Now we have polymerase chain reaction, also known as PCR. So PCR was first discovered by Carey Mullis in 1980s. This technique revolutionized the biological sciences as this method is very fast and accurate. You can make millions or even billions of copies of your gene in just few hours. So how PCR work is it basically uses DNA polymerase to synthesize new strand of DNA. So in this method, the key ingredients you required are the target DNA, your template, which you are going to provide. Then you need primers. Primers are basically short DNA sequences like 20 to 30 nucleotide base pairs, which are very specific and they're specifically designed to attach to a certain part of your DNA. If you have, let's suppose you have a BRCA gene, and you design a primer according to that gene, so that out of your whole genome, these primers can only going to attach on that specific gene, not anywhere else. We will also discuss the designing and selection of primers in the next video. Next you need is DNA polymerase. In this reaction, we need a heat-resistant DNA polymerase because this reaction carried out at relatively high temperature, and the normal DNA polymerase that are present in our body is very heat sensitive and it can denaturate at such high temperature. So we basically use tag polymerase in this PCR reaction. Next, we need DNTPs, deoxyribonucleotide triphosphates. As for the DNA, we need adenine, guanine, cytosine, thymine. These four nucleotides are very important for the synthesis of DNA. We also need MgCl2, magnesium chloride, which act as a cofactor and help DNA polymerase to carry out the reaction. So what are the key steps of PCR? It consists of three main steps. First of all, denaturation. You break down your DNA strands. You have hydrogen bondings. You break down these hydrogen bonds and separate the double helix DNA strand. And this step is known as denaturation. The next step is annealing. Now, your primers, which you have specifically designed, these primers gonna attach to that specific position. The next step is extension. Now, your DNA polymerase, which is a complex, it comes and it run along your DNA strand and add the complementary bases. Like if you have adenine here, it's gonna attach the thymine, and this way the template which you have provided to the reaction is duplicated by the reaction mixture. Now we are deeply looking into each step, what actually is happening inside the PCR tube. You add all the ingredients inside the PCR tube, and then you put that PCR tube into the thermocycler, which is the machine adjustable to different temperature. First of all, you give your template. In the first step, what actually is going to happen is this machine gonna increase the temperature up to 95 degree temperature. This temperature is very high. So at that temperature, the hydrogen bonds between the two DNA strands will break down and this double-stranded DNA will convert into single-stranded DNA. So here you have single strands of DNA separated to each other. Now, the next step is annealing. Annealing temperature varies from 45 to 65 degrees depending upon your primers. So as I have already told you, we will discuss the primer designing and its TM temperature in the next video. Now your primers will come and they will gonna attach to the flanking regions, which are extra adjacent regions next to your gene of interest or your preference sequence. Primers will come and attach at this lower temperature. When we cool it down, cooler reaction, this temperature is feasible for your primers to come and attach to your template. These primers will come and attach complementary to the 3' end. One is the forward primer and one is the reverse primer. Once the primers are attached, the next step is you will increase the temperature up to 72 degrees. At this temperature, your DNA polymerase will come and attach next to your primers. And this DNA polymerase will run along your template and it will attach the nucleotides complementary to your stand, your base, your template. For example, as in this given reaction, you have adenine, thymine, cytosine, guanine, you have all the DNTPs in your reaction mixture. So you have primers, you have DNTPs, everything you have added into your reaction mixture. Once the polymerase attached to the specific position, it will take the complementary, let's suppose thymine complementary to adenine, and it will gonna attach the complementary nucleotide to your template stand. And this way, it synthesizes the new DNA strand towards the 3' end. And at the end of this reaction, you have two DNA strands, two double-stranded DNAs. In the start, we have one strand, one double helix, and now you have two double helix. This way, we can duplicate our DNA by using this reaction. So we were talking about millions or billions of copies, but this is only two copies of the DNA. But actually, it happened. This cycle repeats. It repeats to multiple times. You can repeat it to 33, which are the ideal condition, 33 cycles. So these cycles can be varied. Basically, at this stage, the thermocycler again increased the temperature up to 95 degrees, and once the temperature increase again, the bonding between these two strands break down and they will produce 4 single-stranded DNA. And now the same process will repeat, and this way your single template will replicate into multiple copies. Now, one more question you might be thinking about is, do we need to add more ingredients into the reaction? No, you don't. Once you start the reaction, you add all the things you needed into your PCR tube. Now you have DNTPs, you have primers, you have everything. You just need to change your temperature. By changing the temperature, each step carried out one by one, and this process continue until you stop the reaction. So this way you can amplify your gene of interest and make millions of copies in just few hours. That was all about PCR and how it works. If you like the video, please hit the bell button and stay tuned for the future videos. Please subscribe my channel and do comments about the topics you want to study with me. I would love to help you guys. Yes, Rio? Yes, we would love to help you guys.